Hi everyone, this is Group 2. We are going to talk about thalassemia today.
Let's begin with the introduction. So what's thalassemia? It's an inherited blood disorder caused when the body doesn't make enough hemoglobin. This will reduce the production of normal hemoglobin, and red blood cells will not function properly and they will last less than 120 days. So there are fewer healthy red blood cells traveling in the bloodstream. This will reduce oxygen supply to cells throughout the body and finally cause mild or severe anemia. And there are two types of thalassemia, alpha and beta thalassemia. We will discuss them in detail in the coming slides.
In the first part, we will talk about the heredity factor.
Production of alpha globin in chromosome 16 requires two genes from both pairs, while production of beta globin in chromosome 11 requires one gene from both pairs. Both alpha and beta thalassemia are result from the mutant gene of the globin.
To talk about the changes in molecular level, we can divide thalassemia into two types. The first one is deletion type, which refers to the loss of whole segment of globin genes, and it occurs commonly in alpha thalassemia. And the second one is non-deletion type. It means there's a change in nucleotides within the genes, and it occurs commonly in beta thalassemia. In alpha thalassemia, there are many types of deletions, and the major cause is because of the crossing over, which result in the removal of single alpha globin genes, and it's very rare that a deletion of large segment happens. If we take a look at alpha 1 and alpha 2 genes, we'll find that they are within two highly homologous duplicated segments, and these segments are divided into homologous subsegment XYEZ, and XYEZ box play a part in the crossover. The figures here show you the misaligned chromosome during crossover, which produce a chromosome containing 3 alpha globin genes and another one containing only 1 alpha globin genes. As mentioned, non-deletion type refers to the change of nucleotide bases within the genes, and the change of DNA sequence will downregulate the beta globin genes via the stage of gene expressions. There's many types of point mutation in beta globin genes as well, and most patient genes are having problems in their third exons, and below show you some mutations examples in beta globin genes.
Alpha thalassemia happens when one or more genes that control the making of alpha globin is absent or defective. All mutant genes may inherit to the next generation. It can cause anemia ranging from mild to severe. Most serious form is the alpha thalassemia major, which will cause stillbirth. This example can show how the alpha thalassemia inherited to the next generation. The affected child has two inherited mutant copies of alpha globin gene from mother, so the child will have alpha thalassemia minor. For the silent carrier, they only have one damaged copy of hemoglobin alpha 2, so they are silent alpha thalassemia carrier. This example also shows how the alpha thalassemia inherited to the next generation. If both parents are alpha thalassemia minor, their offspring will have 50% getting alpha thalassemia minor, 25% getting alpha thalassemia major, 25% becoming normal people without thalassemia. Beta thalassemia happens when one or more genes that control the making of beta globin is absent or defective. All mutant genes may inherit to the next generation. It can cause anemia ranging from mild to severe. Most serious form is the beta thalassemia major, which have two mutant genes, which can cause severe anemia. This example can show how the beta thalassemia inherited to the next generation. If both parents are beta thalassemia mild, their offspring will have 50% getting beta thalassemia minor, 25% getting beta thalassemia major, 25% becoming normal people without thalassemia.
In the following slide, we'll talk about the environmental factor. We often hear that there's no direct relationship between thalassemia and the environment. However, many researchers have shown that malaria endemicity is highly correlated with the thalassemia distribution. Here's the distribution map of malaria and thalassemia, and you'll find the affected regions are overlapped. Thalassemia protects malarial patients from a range of clinical manifestations, and in general, protection is greatest against severe malaria, and there's no evidence shown that thalassemia can against the infections. Anyway, people with thalassemia has a greater chance of survival in the region of malarial endemicity, and therefore we will find more people with thalassemia in those regions.
In the following slides, diagnosis and treatment will be discussed.
There are several ways to diagnose thalassemia. First of all, blood tests with complete blood counts can be used to investigate the actual number of red cells and amount of hemoglobin in the blood sample. By calculating the mean corpuscular volume and mean corpuscular hemoglobin, we can measure the average size of red cells and average amount of hemoglobin in a single red cells respectively.
Thalassemia is not rare among Hong Kong infants. 4.3 of the infants are alpha thalassemia carriers, while 2.8 of them are beta thalassemia carriers. Antenatal thalassemia screening targets all pregnant women with antenatal booking. The screening could identify the couples at risk and detect the severe forms of thalassemia in fetus in early pregnancy. Blood tests with complete blood count is used, and usually mean corpuscular volume or MCV will be the parameter. If the MCV of patient is lower than 80 femtoliter, he or she is suspected to suffering from thalassemia. When the MCV of pregnant woman is lower than normal, the father of pregnancy should also receive the blood test to clarify whether the infant was suffering from thalassemia or not. Unfortunately, the thalassemia carrier couples with result positive have 25% chances of giving birth of offspring suffering from thalassemia major.
The second method is blood smear, a thinning of blood that is treated with a special stain on a slide under a microscope to see whether the red blood cells are normal. Patients with thalassemia will have red blood cells that are smaller and paler than the normal one. They are nucleated and have uneven hemoglobin distribution. The third method is iron studies, which can indicate whether the cause of anemia is iron deficiency or thalassemia. Hemoglobin electrophoresis can also diagnose beta thalassemia and can be used for newborn hemoglobin screening and prenatal screening. The fifth method is DNA analysis that is used to analyze the mutations in alpha and beta globin producing genes. Besides, genetic testing can also be used to diagnose alpha thalassemia by taking a sample of amniotic fluid or tissue from placenta. Family genetic studies can be used to see whether there are any missing or altered hemoglobin genes. Individuals with mild thalassemia are not required for treatment. In order to treat moderate and severe thalassemia, blood transfusion is the standard method. With intravenous injection of normal red cells, normal levels of healthy red cells and hemoglobin will be restored. The advantages of blood transfusions are the increase in oxygenated blood supply, prevention of growth abnormalities, and organ damage. However, blood transfusion is expensive and has a risk of carrying infectious agents into the body. Frequent blood transfusions may also lead to a build-up of iron in blood, which can damage liver, heart, and other organs. Another treatment is iron chelation therapy. This involves the administration of drugs that binds with the excess iron and flush it out of the body through urine. The Thalassemia and the oils are the examples of drugs. The third treatment is nutritional supplements. By increasing folic acid supply to patients, vitamin B level will increase to help build healthier red blood cells. The last treatment that I want to introduce is the bone marrow and stem cell transplant. It's the most effective treatment of beta thalassemia major. Patients will receive transplant from a compatible related donor, usually is a sibling, having the same types of human leukocyte antigens on their cell surface. Within 1 month, the transplant bone marrow stem cells will start to make new, healthy red blood cells. However, it is not recommended for those with mild or moderate thalassemia due to its risk.
Rare co-occurrence of beta thalassemia and pseudoxanthoma elasticum, or PXE, is a novel research published on January of 2020. Briefly, PXE is a rare inherited and progressive disorder caused by the mutations in ABCC6 transporter gene. It will result in mineralization of elastic fibers. It is hypothesized that the PXE-like manifestations in beta thalassemia patients are the consequence of oxidative stress-driven epigenetic regulatory mechanism causing ABCC6 downregulation. In the research, 3 unrelated beta thalassemia females with PXE-like manifestations are observed.
In targeted exome sequencing, we analyzed on a panel of 19 genes in total, in which 12 of them are responsible for the calcification-related disease and 7 of them play a role in calcium phosphate equilibrium. In this technique, we perform a variance discovery and a functional annotation. In the research methodology, we use filtering of genetic variants and the in silico analysis for the functional predictions of variants. We also use the Sanger sequencing to confirm all the pathogenic variants and the sequence of the purified PCR products. The research results suggested that the rare sequence variants were detected in the ABCC6 and the EMPP1 genes. They are both PXE-causing genes. However, an ABCC6 pathogenic allele is associated with a new rare sequence variant in EMPP1 gene. So, to conclude, the ABCC6 gene is related to the PXE and the beta thalassemia. The first future development is the beta thalassemia gene therapy with the use of induced pluripotent stem cells. In this technique, we use the CRISPR or the Cas9 technology, and the corrected cells could produce hemoglobin after the therapy. The second one is the ex vivo expansion of the hematopoietic stem cells for the cell and gene therapy manipulations. In this therapy, we co-culture the HSCs with the normal stromal cells. This can preserve the HSC pluripotency, and thus it is beneficial for the expansion of the corrected HSCs. The third one is the ex vivo selection and optional suicide gene of the genetically modified HSCs. In this therapy, we fuse the therapeutic genes with the selection and suicide genes. This can maintain the effective levels of the therapeutic gene, which is very important for the therapy. The fourth therapy is the multiplex lentiviral gene therapy. In this therapy, we coordinate the beta-globin expression and the alpha-2 globin reductions. This requires a lower vector copy number of the viral vector for equivalent efficacies, which can improve the safety for the patients in the therapy.
To conclude, thalassemia is caused by gene mutations, and there are many ways to treat and diagnose thalassemia.